All right, let's start with a sound check if we could. Can you hear me in the back row there, Vern? Can you hear me in the back row? Can you hear me in the back row back there? Excellent. After my frustrations on Wednesday trying to show you that video, someone kindly pointed out to me it's also available through YouTube. And so I thought I would open up YouTube, and YouTube also uses Flash. So all I can do is say, do go look for the links in the links thing. It's really a wonderful video. A couple people have told me they've actually gone and looked at it. It's really great fun. Are there any questions about the overview of metabolism before we start specifically speaking about the metabolism of carbohydrates? Okay. I have on the front page of this a piece of folk biochemistry I thought you might enjoy. The more you learn in this course, the more you're going to be able to read some of the things that are out there with a skeptical eye. But this is particularly egregious. Does anybody recognize what they are talking about when they talk about a dipole, dibase delivery system? Water. Right. It's a dipole, dibase. Right. And the idea that when you dissociate water molecules, you have nascent oxygen that's going to be better for you. I don't know where to begin and how many mistakes there are in that. Anyhow, I thought you'd enjoy it. But let's talk real science. We're going to be talking about the metabolism of carbohydrates. Oops, let me get back there, get the pointer going. And the options we have in carbohydrates for what they can be used for and how we do them. Carbohydrates are actually extremely flexible in the kinds of things that they get used for by the body. So they get used for energy, obviously. Major one there. Short-term glycolysis can act anaerobically. Longer term, we're going to see we need oxidative respiration to do it, to get the most energy out of the sugar molecule. It makes a good storage molecule. So we store sugar as glycogen in muscle and kidney and liver. Plants store sugar as starch. And so we'll be looking principally at the glycogen part of all that. Sugar is important structurally. Glucose gets used to make cellulose. Cellulose may end up being the fuel of the future if we ever get switchgrass figured out. Glucose is a good precursor, and we're going to see a number of places where that's true, where you can use it to make the ribose sugar for bases. You can use it to make fatty acids if you've eaten too much sugar, which most of us have had that experience. You can use it as a precursor for amino acids. And finally, it is one of the sources for reducing power that we mentioned at the beginning in the overview, that part of anabolic metabolism, the building up aspect of metabolism, is you have to reduce things to make them more interesting. And sugars are one of the major sources for that kind of reducing power. And this is just showing you a couple of organisms that use glucose. So you've got the red blood cells that work off glycolysis with a little malarial parasite there that has both. And here's a yeast cell. And yeast metabolism is kind of interesting because they can do glycolysis and produce ethanol, which makes life interesting. But let me give you a wide-angle view of where we're going to be going so you have a sense of what we're going to be talking about pathway by pathway, because there's a number of pathways that are involved in carbohydrate metabolism. We've mentioned briefly that the breakdown of glycogen happens through phosphorylasis. That's a way to get the monomers out of that. We're going to look in detail at the breakdown of the monomers, the glucose, through glycolysis. Glycolysis tells you glyco is something to do with sugars, lysis, breaking down. So glycolysis is for breaking down sugars. We're going to look at how you make new sugars. It's not just plants that can make sugars. We can do it too. And when you look at the name for that, gluco, something about glucose, neo, something about new, genesis, becoming, making, so this is making new sugar, making new glucose. And then we can also make new polymers out of it. And from the small carbon intermediates, we can do a bunch of other stuff as well. So we're going to be talking sort of above the level of citric acid cycle for the next 3 lectures. So the handout that you got today is for 3 lectures. It's the only one that I've done that for. I've done that partly because I want to make sure you have time enough to learn glycolysis. We're starting a chunk of the course now where metabolism is going to be coming into it quite a bit. And it's a different kind of a thing to learn than some of the other things we've been learning or maybe you've ever learned before. And these are some tips I have offered for how to go about learning metabolic pathways. What do you need to do to learn one? And these are some questions you should be asking yourself for each pathway. What is it doing for the cell? What's its function? What's the purpose of it for the cell? What do you put in at the top? What are the inputs, or maybe on the side? What are the products? What's going to come out the end? Does it cost ATP or does it yield ATP? Some pathways will do both, in which case you want to know where it does which. Are there any steps that use oxidation or reduction? Many pathways do have at least one, sometimes several, and they're usually important steps. Okay? How is it regulated? If every pathway in our bodies did everything at once, we would just be a mass of jello. We would be incoherent. And so every pathway has a level of regulation with perhaps one exception, which I'll point out when we get there. And then are there other pathways that resemble it? I mention this because I'll try to sort of remind you of this as we go along. There are a number of places where once you learn this pathway, you know 90% of this pathway. And so it's really worthwhile to learn the first one because you're learning two for the price of one. And we'll point those out as we go along. We haven't yet really talked about carbohydrate structure. And so before we can get into the metabolism, we have to do a little bit of structure talking. And so here's some sort of an introduction to carbohydrate structure. The common feature of carbohydrates is that they are polyalcohols. The name kind of tells you that. It's carbon that's been hydrated, all right? So carbons that have, on average, a water molecule on them. So just about every carbon is going to have an alcohol group hanging off. So they're polyalcohols. Some sugars are linear. Some are in a ring form. Many can interconvert between linear and ring form. And we're going to see when that's useful. Trying to draw sugars has been a challenge to some extent throughout the history of biochemistry, because on the one hand you'd like to represent the molecule as faithfully as you can. On the other hand, the page is flat. And so there have been a number of more or less successful conventions developed to try to make it possible to represent sugars. Here's one that you may have seen already called the Fisher projection, in which we've sort of ironed it out flat, and you can tell the handedness of the alcohol groups by left or right rather than above or below the plane. A linear molecule like this can close into a ring. So this is showing you sort of what's happening. So the alcohol on the 5 carbon is going to attack the first carbon, and you're going to have a new bond formed here between that oxygen from the alcohol and the first carbon. So now you have a ring. And you can see that this first carbon, that carbonyl oxygen is free to rotate around. And so when the ring closes, it could be up or it could be down. And this is one of the first aspects of sugar structure that is important. If it closes with the alcohol down, we call that an alpha anomer. If it closes with it up, that's the beta anomer. They're both glucose. But in one case, it's the alpha because the oxygen's down. In the other case, it's beta with the up. And they differ only at the anomeric carbon, at this one carbon. So anomers are compounds that are distinct from each other because of transitions like this. Okay. They're otherwise the same molecule. It's interesting to look at some of the features of glucose and why perhaps it's such an important sugar. The first two representations didn't really capture the three-dimensional aspects of it. And this kind of projection is a third style of representing it, which is more nearly faithful. So it's trying to capture the fact that it's not a flat ring, it's either what we call a chair or a boat. So if both things are flapping up, that's the boat configuration. If the one's up and one down, it's the chair configuration. And if we compare glucose to cyclohexane, in cyclohexane, all the carbons have on them is hydrogens. In glucose, it has alcohols, but the alcohols are all in the equatorial position. They're pointing away from the center of the ring. The axial positions that go up and down are all hydrogens. And glucose is the only sugar that has all equatorial substitutions when it's in the chair configuration. And this may be important to why it was picked to be the most common, widely used sugar. Now, because glucose comes in two different forms as a ring, either beta or alpha, you can make different polymers out of it. First of all, I should mention that the polymers of glucose all use the ring structure. They don't use linear glucose. They all use ring glucose, which isn't obvious on the face of it, but it's a true fact. If you start off with glucoses that are all alpha, you can make one kind of polymer. If you start off with sugars that are all beta, you can make a different kind of polymer. So if you have alpha linkages, so this one carbon was an alpha when this carbon was added to it. If it's all alpha linkages, we're going to end up with either starch or glycogen. Glycogen is sometimes called animal starch. If that anomeric carbon, the one carbon, was in the beta position, you now have a different packing, and that's called cellulose. Those don't look all that different chemically, but it turns out that the position, the configuration of that anomeric carbon makes a world of difference to the overall structure of the polymer, and we'll see that here. If you have all beta 1,4 linkages, you end up with cellulose, and cellulose tends to line up in parallel molecules, and the molecules are hydrogen bonded across from one molecule to the next, which should sound like another beta thing we've talked about, beta pleated sheets. Whereas the alpha 1,4 linkages are going to give you starches and glycogen, which can be branched. The cellulose is not a branched compound, but starch and glycogen are, depending on how much they are. And we can see that at a level of the subunits here by this diagram that represents each little ring that's sort of a sequin, if you like. So the alpha 1,4 linkages in starch give you a helix of packing. The beta 1,4 linkages give you, that's going to be the next picture. So these are all different examples of the alpha 1,4 linkages. This is amylose and amylopectin. Amylose is the least branched of the starches, amylopectin somewhat branched. Glycogen is even more branched, and so it's looser than the starches. It's because the starches have this helical structure that you can stain it for iodine. Have you ever used iodine as a test for starch, and it turns deep purple? It's turning deep purple because it fits into the center of that helix, where it's in a different chemical environment. The other neat thing about it is that the starch granules over here are basically anhydrous, and so when you heat them up in gravy, they eventually explode and are denatured, and you can imagine that if you had everything packed down tight and now you have those long skinny molecules flopping in the breeze, it would make things thick, and that's exactly where the viscosity of gravy comes from. This is also the precursor of malt for beer, so the maltase that's found in yeasts can be used for this. Here's the cellulose at a little more molecular level. This is, again, the beta 1,4 linkages here. And you can see now how the alcohols sticking out on that equatorial position can make hydrogen bonds from side to side, from one strand to the next one. So this is a lateral linkage. It's a more extended linkage. And it really is structurally very reminiscent of the beta pleated sheet. Because all of the hydrogen bonds are occupied, everything in this packing, all the hydrogen bonds are taken up with cellulose. It makes cellulose pretty insoluble and also very strong. So you wouldn't think it, but it turns out that glucose polymers have secondary structure. They have a primary structure, which is very boring. It's glucose, 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 glucose. Very easy primary structure to remember. And then they have a secondary structure that determines whether they're the alpha 1,4 or the beta 1,4 linkages. It's not really appropriate to talk about tertiary structure because they come together in such a lot of different ways that there isn't one consistent 3D packing. So they have secondary structure that's interesting and different between the two of them. Their primary structures are similar, and they don't have tertiary structure. When you make rayon, you take that cellulose molecules and you can convert some of the side groups into other groups that make it more water soluble, so that you can then pass that through a thin tube, and as it goes through that thin tube, it passes into a chemical that reverts that modification back into cellulose. And so you can regenerate a cellulose fiber, but now it's one that has the mechanical properties that you want it to have, the diameter and so on. That rayon is used for a lot of things. You've probably run into it for clothing, but things like industrial uses are quite prominent as well. We're not as aware of them. Do you think that starch would make a good fiber the way like rayon? You can talk to each other if you want. Would starch be good to make a fiber out of? Excuse me. Sorry, video people. Anyone want to take a stab at whether cellulose could make you a good fiber like rayon? Sorry? Did I hear? Anyone want to volunteer? What do you think? Would it make a good fiber? You don't know. That's an honest answer. Honest answer. Anybody else want to take a stab at it?
I'll say that.
Okay. And we want to take a stab at an explanation too of why no? It's what? I see. Well, with the yes/no answers, you do have 50% chance. It just goes back to this idea of a lateral hydrogen bonding, that the structure of starch doesn't provide that possibility of side-to-side hydrogen bonding. So you can pass it through all the chemicals you want. It's not going to be able to make that side-to-side bonding. It doesn't have the potential for making that kind of strength. People have found ways to make foam out of starch, and you may have had coffee cups out of starch foam. And for that purpose, it's perfectly strong enough. Oops, wrong direction. Oops. Yeah. Okay. I already mentioned that. Oh, this is, no, okay. So let's look at glycogenolysis. We've talked about glycogen as a polymer. It's quote animal starch. It's alpha-1,4 linkages in a branched form. So it's a somewhat looser storage form for carbohydrate, specifically for glucose. Not much use until you get something out of it. So we have to get at that sugar to make it useful. And the process called glycogenolysis is how we do that. And its name tells you what's going on here. So glycogen is a molecule that gen can give rise to sugar, specifically glucose. So glycogen gives rise to sugar. Glycogenolysis is breaking down the molecule glycogen. And we mentioned this briefly before, but it's extremely acute. Glycogenolysis uses inorganic phosphate. And the enzyme that does that is called glycogen phosphorylase. So the name is also telling you that something special is going on. This isn't a hydrolase, this is a phosphorylase. It's using an inorganic phosphate to break a bond, and that phosphate then is incorporated into the monomer that's released. If you'd used water to do it, you'd have a hydrogen or an oxygen from the water molecule. We used a phosphate to break it, so the phosphate is now covalently part of the monomer. So glycogen phosphorylase will use phosphate to create glucose 1-phosphate. Glucose 1-phosphate is not yet useful for other processes, so we're going to have to do a little rearrangement. And the enzyme that does that has a rational name, phosphoglucomutase. So mutase is going to mutate it, move it around. What is it going to work on? A phosphoglucose. So the product of phosphoglucomutase is glucose 6-phosphate. And you notice the double-headed arrows here. This is not a double-headed arrow, this is a single-headed arrow. This is a one-way process with a significant delta G, because that phosphate on the sugar isn't enough to remake a bond. But the phosphoglucomutase has double-headed arrows because there isn't much difference between glucose 1-phosphate and glucose 6-phosphate, not in structure at least. Now, in the context of the cell, we can make it principally go to glucose 6-phosphate by using up the glucose. But that's not due to the enzyme. That's due to the exact context biochemically in the cell. So that this is an example of where the actual free energy depends on the actual conditions. Now, in the liver, I don't think your book quite emphasizes this enough, but in the liver, there's another enzyme called glucose 6-phosphatase, and it can eat off that phosphate and throw it away. And that then provides for a glucose molecule, pure and simple. The liver can do this when it wants to release sugar into the bloodstream. So the liver is the only organ that can store sugar as glycogen and then put it back into circulation. Muscles and kidneys store sugar as glycogen, but they use it locally. Only liver is generous enough to give it back to everybody else. And this is another rationally named enzyme, glucose 6-phosphatase. It's a phosphatase. It takes off phosphates. So we're ready now to start talking about glycolysis itself. We have molecules we can use to do this with. And glycolysis is a very important pathway. It is ancient. It is present in all organisms. And it is something that has been here before there is significant oxygen in the atmosphere. So the first organisms that were alive on Earth had an atmosphere that was neutral or maybe slightly reducing. They had no oxygen for aerobic respiration. They had to do everything anaerobically, and glycolysis was the trick that they used for it. I said nearly universal because there are a few interesting exceptions, but it's certainly one of the most widely spread pathways. Overall, what we're going to try and do is turn one 6-carbon glucose into 2 3-carbon pyruvates. You're going to find the pathways easier to keep track of if you keep track of your carbons. Carbons are never created or destroyed in biochemistry. You have to go to the nuclear physics department for that. We're not just doing that for idle purposes. We're going to produce 2 ATP in the process, and we're going to reduce 2 NAD to 2 NADH. We're going to see that there are 2 phases to glycolysis. There's an energy investment phase where we put energy into the processes, and then a second phase where we take energy out. In addition to being part of the key part of the energy metabolism, glycolysis is also a fertile place to get intermediates for other things as well. And we're going to see what kinds of things when we get down there. So let's start taking a bit of a look at this. Another tip for learning metabolic pathways is to take those dry erase boards, you can dry erase, and draw the first compound in the pathway. And then you play enzyme and go in with the eraser and erase the bit that's going to get changed and draw the new bit. So if you play enzyme on the dry erase board, it will help you focus on what is happening to the molecule and the logic behind it. Is it getting rearranged? Is it getting reduced? Is it getting oxidized? Does it get a phosphate? And so on. So playing enzyme with a dry erase board can be very helpful. So let's take a quick look at this overview. If we start off with glucose in this case, we can turn that into a usable form for the glycolysis with an enzyme called hexokinase. That's going to cost us ATP. And now we have a phosphorylated sugar, glucose 6-phosphate. So here's the plain glucose here, just a bunch of alcohol side chains. Here's glucose 6-phosphate. There's the phosphate sticking up on the 6-carbon. Glucose 6-phosphate is then a substrate for an isomerase, and it can turn glucose 6-phosphate into fructose 6-phosphate. These are both 6-carbon sugars. Glucose 6-phosphate's the 5-membered ring. Fructose 6-phosphate's a 6-membered ring. So now the 1 carbon is sticking up here like a flag. The The fructose 6-phosphate is a good substrate for another phosphate to come in. So an enzyme called phosphofructokinase. So it's a kinase, so it adds phosphate. It adds phosphate to phosphofructose. So phosphofructokinase is the enzyme that takes that substrate and generates this sugar that has a phosphate flapping in the breeze off both sides, which is fructose 1,6-bisphosphate. So far, we've invested 2 ATPs and have nothing to show for it except a highly charged sugar molecule. This molecule, the fructose 1,6-bisphosphate, is a good substrate for an enzyme called aldolase. And aldolase can split that into 2 3-carbon fragments, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So here's a picture of glyceraldehyde 3-phosphate, and here's dihydroxyacetone phosphate. In dihydroxyacetone phosphate, the carbonyl oxygen is in the middle. In glyceraldehyde 3-phosphate, the carbonyl oxygen is on the end. And they both have a phosphate hanging off of them. What we really want to work with is the glyceraldehyde 3-phosphate, and there's an enzyme that can convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate without any investment of energy. You'll notice that some of these steps are one-way arrows and some of them are two-way arrows. So when we invest ATP here and invest ATP here, those are both one-way arrows. They're essentially irreversible reactions. On the other hand, if we're rearranging glucose into fructose through an isomerase, that's a two-headed arrow. If we're, and interestingly, if we're splitting fructose 1,6-bisphosphate into 3-carbon fragments, that's also a double-headed arrow. So that has actually a reversible reaction, as is, of course, the conversion between those two. All right. So far, all we've done is to add phosphate to the molecules and break it into 3 bits. What the phosphate did on the carbon, on the sugars, and then on the 3-carbon fragments, first of all, it kept it inside the cell. Remember when we were back talking about membranes, about passive transporters, if the gradient is higher inside the cell than outside the cell, you'll leak it out and lose it. But if you phosphorylate every sugar as soon as it comes in, it won't leak back out because the phosphate keeps it from going through the channel. So that's one important thing, is the ATP is capturing the glucose to keep it in the cell. But it's also setting up that carbon backbone for the future reactions. And the second half of glycolysis is where it starts to pay off with anything. And it does some fairly interesting things along the way. So we made the dihydroxyacetone phosphate into a glyceraldehyde 3-phosphate. So now we have 2 molecules of glyceraldehyde 3-phosphate. And the enzyme glyceraldehyde 3-phosphate dehydrogenase is going to work on that. It's a long name, but these things will start to make patterns for you, where the enzyme names will start to make sense. It's a dehydrogenase. You can bet your bottom dollar electrons are involved. And because it's a dehydrogenase, we now are going to have a reduced NADH, right? The NAD has been reduced to NADH. And then the substrate for it is just the molecule we've been working on so far, glyceraldehyde 3-phosphate, or GA3P sometimes it's called. But it's an odd reaction because it's going to consume inorganic phosphate and NAD plus to release a bisphosphate glycerate. So this now has a phosphate on this end and a phosphate on that end. So this is a bisphosphoglycerate and reduced NADH. Odd reaction. We're going to look at that in more detail. Because this molecule has phosphate on both steps, it's set up in a way now that it can provide a high energy bond to something else. So phosphoglycerate kinase comes in and generates ATP from it. This enzyme is named for the backwards reaction. It's named as if it's going from here to there. In glycolysis, it goes from there to here. One of the few enzymes named for the backwards reaction. Now we have 2 molecules of 3-phosphoglycerate, and you can see that it's a glycerate up there now. It's not an aldehyde, it's a carboxylic acid. So that's evidence that that carbon got oxidized, right? So we have three phosphoglycerate. We have another mutase, which is going to do a little rearrangement for us and make a 2-phosphoglycerate. So here's a molecule, 2-phosphoglycerate, 3-carbon backbone, carboxylic acid there. This is a double-headed arrow, so you can imagine that's highly reversible. But what's interesting is that by moving the phosphate to the middle here, we've set up the possibility for another reaction, a dehydration reaction. So if you take a water molecule out of that, you can generate a molecule called phosphoenol pyruvate. And so if you look at this, it's a phosphoenol. There's an ene, a double bond there, and that would be an alcohol if it didn't have a phosphate on it. So it's a phosphoenol pyruvate. And that strained-looking thing also is a high-energy bond, and so you can use it as a donor to make ATP. And the product of that would be then ATP and pyruvate. This is also named for the backwards reaction. In glycolysis, it's going from the phosphoenol pyruvate to pyruvate. The enzyme is called pyruvate kinase, as if it went backwards. Again, if you set things up in the lab, you can make these things run backwards if you put in enough ATP, but in the cell, the way it's going is from phosphoenol pyruvate to pyruvate. So we've actually recovered all the ATP we invested, right? Because we got ATP out here, and we got ATP out there. And we've got some NADH, which could be interesting, might not be interesting, depending on whether there's some oxygen around. So at least we made a little profit. Not a huge amount, but we have made a little profit. Okay. So that's the overview. That's kind of the Cook's tour. We're going to talk about each of those reactions in a little bit more detail as well. I want to encourage you to learn glycolysis quickly, not try to wait until the test downstream, because if you learn it in the next few days, you're going to have it in your mind when we do the rest of the things. If you put off learning it till later, it's just going to be that much harder to learn the things in between. So please start thinking about it soon. That's one of my reasons for spreading carbohydrate metabolism over 3 lectures, is to make sure you have enough time to learn it en route, not later on. So let's look at hexokinase in a little more detail. It is the beginning of the hexose phase when you have 6 carbon sugars. 1 ATP consumed, and this is the 6th carbon position there. Hexokinase is like a clamshell. It is in an open position to begin with, and then it closes down to cover the substrates. This is a substantial free energy change, negative 17 kilojoules. So this is a strongly one-way reaction under any metabolically interesting conditions. So if talking in terms of what will happen in the cell, this only goes one way. I've mentioned that the substrate is now going to stay in the cell because it's charged. That phosphate is also one of the things that will bind to like the substrate binding aspects of enzymes. So when it's going through different reaction steps, the phosphate is always a salient part of that. Depending on where you get your glucose from, it can be more efficient or not. If you get the glucose from the bloodstream, you've got to invest in ATP. If you got your glucose from glycogen, you did that through phosphorylasis. And phosphorylasis does not cost an ATP. So glucose coming from glycogen has a higher energy yield than glucose out of the bloodstream. Any questions about that? Okay. Next step is a fairly straightforward one. Here's its free energy change, very close to neutral, freely reversible. What's actually going on here is we're changing the organization of the molecule here in the first carbon. So glucose 6-phosphate is a 6-membered ring, fructose 6-phosphate is a 5-membered ring, or in the Fischer projection, glucose is an aldose, meaning an aldehyde up here. Fructose is a ketose, meaning it has a carbonyl carbon there. So in a fructose, the carbonyl carbon is interior. In glucose and other aldoses, the carbonyl carbon is at the end. Phosphofructokinase, big enzyme, important one. This is a good one to remember. It's going to take fructose 6-phosphate, with the phosphate flapping in the breeze on the 6, and spend an ATP to phosphorylate it at the 1 position. So now you have both the 1 and the 6 carbons, and you're releasing an ADP. Again, minus 17 kilojoules per mole. This is a very strongly favored reaction. Phosphorylation reactions often are, sometimes not. There's a couple in glycolysis that aren't. But in terms of this reaction, it's favored that way. Phosphofructokinase is very highly regulated. They're going to be, when we talk about the regulation of glycolysis, this is going to be a target for a lot of regulation. And the reason it is we call it a committed step. That fructose 6-phosphate can be used for other things than glycolysis. It can easily go back to glucose 6-phosphate and then maybe go off into reducing power production or go off into glycogen storage. Once it passes through phosphofructokinase, it'll probably complete the rest of glycolysis. So this is a committed step. And we're going to see frequently that the committed step is a regulated step. And so some of the kinds of regulation here make a lot of sense. So for instance, ADP is a product of this. ADP being around will facilitate the binding of the substrate. It'll also increase the activity of that enzyme. So ADP is a relevant and meaningful signal that energy is needed. Phosphoenolpyruvate is an end product of glycolysis. It isn't of this reaction, but of the whole pathway. And phosphoenolpyruvate will reduce binding and activity. It's a signal that the end product is accumulating. We don't need this part of the pathway to keep going very much because we've got all we need. Those are both true facts, and I don't want to diminish them, but in us, in mammals, another regulator was found in fairly recent time called fructose 2,6-bisphosphate. And we're going to talk a little bit more about that. So the substrate, or the product of the enzyme, is fructose 1,6-bisphosphate. A close analog, fructose 2,6-bisphosphate, will turn out to be a regulator. Here's just sort of a tapping into some of the kinds of things that can regulate phosphofructokinase. You can see why these molecules would be things that would increase activity. ADP, GDP, those are both lower energy than ATP or GTP. AMP, an even more low energy form of it. Fructose 2,6-phosphate is a positive regulator, but it is going to be regulated through hormones and at the level of the organism, not at the level of the local environment. We can also turn things down. Phosphoenolpyruvate we talked about as an end product. Citrate can have a negative effect. Citrate is part of the citric acid cycle. If there's a lot of it around, that's also a signal that you've got plenty. Fatty acids, an alternate energy source, that can turn it down. And ATP, a more immediate sign that energy needs are sufficiently satisfied at the moment. So this is, you can think of phosphofructokinase as a kind of a molecular computer. It's along the level of the complexity of hemoglobin, except more so. And you could really learn a lot of enzyme kinetics by just thinking about phosphofructokinase. It's not just that ATP changes activity, it changes the kind of activity. So in the low ATP conditions, or when you have AMP around, it will have a hyperbolic curve. If you look at its activity at a higher level concentration of ATP, it now has become sigmoidal. So at this particular concentration of ATP, you haven't changed the Vmax in particular, but what you have made is that at the lower concentrations of phosphofructokinase, the activity will be much, much lower. The ratio between AMP and ATP is kind of a measure of how much energy is available to the cell. And AMP is a better measure than ADP because if you have two molecules of ADP, you can make one ATP out of that and an AMP. So that's one key measure for it. Protons can inhibit the activity of the enzyme, and so you're not going to be able to do anaerobic exercise to the point where you're poisoning yourself with acid. I mentioned already that citrate is an inhibitor. Here's a picture of the enzyme, and you can see here multiple catalytic sites. So there's a catalytic site, here's a catalytic site, and then there are allosteric sites elsewhere where these things can bind. So ATP can bind at the catalytic site as a substrate. It can also bind at an allosteric site and be a regulator. The kind of regulation that ATP does here is not simple competition. It's an allosteric regulation. Okay? Any questions about that? Okay. So, here we're talking then about, this is sort of a schematic of the regulation that we talked about already, about phosphatidylpyruvate slowing it down and ADP acting as a stimulant. In mammals, we have a different molecule available. So there's an enzyme called phosphofructokinase 2. This is the second one that was discovered, and it makes a fructose 2,6-bisphosphate. So the phosphate is hanging down here now instead of up there. And the fructose 2,6-bisphosphate, or F26T, is going to bind to an allosteric site to alter the activity of the enzyme. And it is a positive regulator, a positive effector of enzyme activity. The levels of activity of this enzyme, phosphofructokinase 2, or PFK2, are under hormonal control rather than just local control. And just how dramatic that fructose 2,6-bisphosphate can be should be clear from these slides. So in the absence of any fructose 2,6-bisphosphate, this red line is an indicator of the activity. So we're raising substrate concentration here, fructose 6-phosphate. Here's the relative velocity of the enzyme. And a very small amount, this is 1/10 micromolar fructose 2,6-bisphosphate, has changed the Km a lot. Let's assume that the Vmax in this state is the same. The Km for this is about here, is down around close to 2 micromolar. In the presence of 10th micromolar fructose 2,6-bisphosphate, we've pushed that down to maybe 10th micromolar or 2/10 micromolar for the fructose 2,6-bisphosphate. So, and 1 micromolar F26P even more so. So this radically changes the affinity of the enzyme for its substrate, fructose 6-phosphate. Fructose 2,6-bisphosphate can also overcome inhibition by ATP. And so here's an enzyme activity curve with ATP, which shows you just how strong the influence can be of ATP. You need it around to be a substrate, so there has to be some rise in activity with it. But it doesn't take a great deal of ATP for it to start to be inhibited and turn the enzyme back off. Here we have that 10th micromolar fructose 2,6-bisphosphate, and now enzyme activity, the peak is a good deal higher, and it continues to act up in a higher concentration of ATP. So major changes to the kinetics. Here with 1 micromolar, higher peak still, and even greater alteration to the sensitivity as a substrate. So it can increase affinity for fructose 1-phosphate, which is one of the substrates, and it decreases the sensitivity to ATP when it's acting as an allosteric regulator. Fructose 2,6-bisphosphate is not an intermediate. It is not a metabolic intermediate. It's not going to get used in any pathway. It's made for this purpose, and then it's broken down. And it's made by an enzyme that has both a kinase domain, which can make fructose 2,6-bisphosphate. It also has a phosphatase domain, which can break it down, and then a large regulatory region. So one enzyme complex has the capacity to turn on or to turn off phosphofructokinase through this allosteric regulator. Any questions about that before we move on to the next bit? Kind of a funky enzyme. So this is fairly complex kinetics, and it gives you kind of a feeling for how important phosphofructokinase has to be to invest this much effort in it. Okay. So now we've got our molecule of fructose 2,6-bisphosphate, and we're going to look at aldolase. So it's going to take one 6-carbon sugar, which has phosphates on both ends, and split it into 2 trioses. Since they're 3-carbon sugars, they're trioses. Dihydroxyacetone phosphate, and you can see how that gets its name. The carbonyl oxygen there would make it acetone. This is hydroxy. There was one there before we phosphorylated it, so it's dihydroxyacetone phosphate. The other 3-carbon piece is the glyceraldehyde 3-phosphate. There's the aldehyde group. This was glycerol, and now there's a phosphate on the 3 position. So glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. So it's the reverse of an aldol condensation. In other words, it would, when you go in this direction, it would be a condensation reaction. In the cell, it has a free energy that's negative because the products are used so quickly. This is an example where the metabolic conditions in the cell account for switching the sign of the free energy change. But under delta G0 prime, it would have a 23 kilojoule per mole drop. But the trioses are used up so quickly that the actual free energy change, the delta G that's actually occurring, is actually less than 0. Dramatic difference because we're pulling out these products and favoring the reaction that way. And I think we'll hold off on this enzymology till Monday. Too late in the day to think about enzyme mechanisms.